Hello viewers, in my last lecture in the series of immunology, I talked about immune cells and those were the granular ones. So last lecture was about granular leukocytes. Leukocytes, since they do not have any pigment, they are also called WBCs. Now coming to agranular ones are of two, they can be, okay, first we will talk about their morphology. They lack, they do not have, or we can say they do not have visible granules. So they lack granules in their cytoplasm, and so their cytoplasm looks very clear and their nucleus is visible. Because of clear cytoplasm, nucleus is visible, which is quite large, and it's not lobed also. And they are also referred as mononuclear leukocytes since they have just one nucleus, which is also a large one, which is different from granulocytes because granulocytes were multi-lobed and not one big nucleus was not present in them. In case of agranulocytes, one big nucleus and granules which are not very visible, so that they are called agranulocytes because they do not have very visible granules. Agranulocytes are further divided or can be studied under two heads, monocytes and lymphocytes. Now, what are monocytes? Monocytes can be spherical in shape, amoeboid in shape, like this. Monocytes, this is a big nucleus monocyte is having. There is no granules are not visible in it. And these monocytes too are the police against enemy. So wherever there is a signal, they sense in the environment there is some enemy. These monocytes rush like other leukocytes. They rush to destroy the invaders. And these monocytes are roaming in the blood, and from the blood, when they are the precursors of two cells, if from the blood they reaches to tissues, there they are named as macrophages. And in lymph nodes, the police is sitting at different lymph nodes, and then there they are called dendritic cells. These dendritic cells and macrophages both are antigen presenting cells. They catch the enemy and they present it to the cells who are going to take action. So they are the initial soldiers who will come and catch the enemy. The macrophages are in the tissues, dendritic cells at different locations. Function of both is to present the antigen to T cells. Later on, we will be reading about T cells, but remember, these two cells are very important antigen presenting cells, APCs. And like eosinophils, which were granular leukocytes, these monocytes too play a role in facilitating healing and repair after the fight. So they are the ones who will repair the tissues after the fight. Their number is 5 to 10% of total circulating RBCs, total circulating WBCs in healthy individuals. So they are 5 to 10%. Monocytes do have subtypes, and the subtypes of monocytes are classical monocytes, non-classical monocytes, intermediate monocytes. Monocytes are spherical in shape with one large nucleus. This is the basic character of every monocyte. Now, they are divided into three subtypes. What is the difference in these three subtypes? First, let's talk about classical monocytes. Classical monocytes, in that percent of 2 to 5% of total number of monocytes moving in the blood cells, 80 to 90%, 95% of total monocytes is the population of classical monocytes. So classical monocytes are maximum in the number in three subtypes, which we will be studying. First is classical, which is maximum in number. How it differs from other subtypes? Because of the presence of CD14. CD is short form of cluster of differentiation. What is this cluster of differentiation? Cluster of differentiation is a marker. A marker protein. Marker protein is a receptor protein. receptor who is looking for that kind of antigen which can enter and which can fix on that receptor. Usually LPS kind of antigen, lipopolysaccharide kind of antigen, which is having lipopolysaccharide wall around it, is the one which is going to be caught by these receptor molecules. Once this antigen anemia is caught by this CD14 marker, it will binds to the LPS, that is lipopolysaccharide antigen roaming around. CD14 marker is there. It will be having a LPS binding protein in it, and this LPS binding protein will bind to the antigen, LPS antigen. Then when this total binding will take place, CD14 molecule having a receptor for LPS, that is LPS binding protein also there. These will fix the antigen, and this fixing, once this key and lock arrangement takes place, then the opening of what happens, this will open or activate cytokines and chemokines, and these cytokines and chemokines will further help in destroying the invaders. So this is the role of classical monocytes. Coming to non-classical monocytes, which are less than classical, that is 2 to 11%, lot less than classical ones. They are only 2 to 11% of the total monocytes. And the difference between classical and non-classical is this non-classical ones express cluster of differentiation 16 marker, CD16 marker. This CD16 marker is a receptor which, when get information, it will activate when the CD16 marker will catch an enemy. This lock and key arrangement will activate natural killer cells. Now, what are natural killer cells? We will be reading it in later slides of today's lecture only. But just at this point, remember that these natural killer cells are roaming to kill the cells of self. They kill the self cells because they will notice that these cells can, if they are survive, they are going to cause problem to the body's self, in which all the immune cells are present. So these natural cells will kill those cells which can cause problem, like cancer causing cells. It's going to kill cancer causing cells. It also kill virus, those cells which virus infected, those cells which have got virus infected in our body. These natural killer cells will kill those cells too. So vaccination works on this natural killer cells. They activate the natural killer cells. This we will be reading in more detail. And non-classical monocytes, because of their CD16 marker, also maintain immune hemostasis in both T cell and antibody dependent signaling pathway. Antibody dependent signaling pathway is something which we are going to read in later lectures, because it's a big thing and I will explain everything. And still I haven't delivered a lecture on antigen and antibody, which is very important. You should know, when we call any all foreign particles are not antigens. Like an apple seed which we eat and it gets destroyed by our stomach juices, then it's not an antigen. It's just cleared by our stomach juices itself. But when we are going to call any foreign particle as antigen, that I will be talking also in my next lectures. I'm trying my best to be as simple as possible. Immunology is not very simple, but I'm trying to make it as simple as possible. Now, I was talking, let's come back to the topic. I was talking about monocytes, and 3 subtypes I have talked. One is classical one, another one is non-classical one. And basically they were classified on this basis because of the presence of a protein molecule, which was cluster of differentiation molecule. And in case of classical one, CD14 was present. And CD14 is when CD14 is like a lock looking for the invader key, and with this lock and key, when proper key fits in a proper lock, then it activates chemokines and cytokines, which is going to kill the invader. In case of non-classical ones, the CD16 was there, and CD16 was activating natural killer cells. And then comes third subtype is intermediate monocytes. Intermediate monocytes account for 2 to 8%. They are the least in number, and they can express both. As the name signifies, they can express CD14 or CD16 both, and CD16 both. They can express both. And I have already told you the role of CD14 and CD16. Coming to the second type of agranulocytes, they are the lymphocytes. Lymphocytes, as the name says itself, they are found in the blood as well as lymph tissues. Lymph tissues are like, they are also found in skin, tonsils, intestines, and in the lining of airways. Their shape is similar, as they are classified in agranulocytes. Their cytoplasm is not very clear. They too have big nucleus and condensed hemochromatin. Their number is 20 to 24% of the entire number of WBCs. Lymphocytes are further, this I have told you, lymphocyte has more hetrochromatin, and which is condensed, so it has a large big nucleus, and this is the nucleus. Nucleus is larger than monocytes, and these lymphocytes, as required, are, because of the different kind of enemy that enters, these lymphocytes get differentiated into B lymphocytes, T lymphocytes, and natural killer cells. So these three are lymphocytes which gets differentiated as and when the signaling and the type of signaling that takes place. Next, what happens when, how these T cells are, what role and how they are going to work further. Let's talk on that. B and T cells origin is same. They both originate from stem cells in the bone marrow. In human beings, I'm delivering the lectures on immunology, it's totally focused on human beings. I'm talking immunology that takes place inside a human body, So when coming back to B and T cells in human body, they all originate from stem cells in the bone marrow. Now, some of these stem cells migrate to thymus, so they mature there in the thymus, so they are called T cells. Cells, lymphocytes, which mature in thymus, get educated in thymus, are called T cells. And the cells which remain, which are not going, lymphocytes which remain in the bone marrow, they develop into B cells. They have morphology, different morphologies. They develop different morphologies. When we call T cells, then they have a receptor on cell membrane, which is referred as T cell receptor. T cells are very important cells, and they play a major role in cell-mediated immunity. What is cell-mediated immunity? How it takes place will require another lecture. That I will take another lecture. I'm trying to be as simple as possible. These T cell receptors, these T cells have T cell receptors, and these T cells multiply and differentiate into 3 different kinds of T cells according to their roles. Some are helper, T helper cells, T regulatory cells, T cytotoxic cells, and some become memory T cells also. Memory based immunity, that we will be discussing in the chapter when we will be reading immunity types. Just remember, T cells can become memory T cells too. So after multiplication and differentiation in thymus, T cells are sent to peripheral tissues, or they circulate between blood and lymph. T cells circulate between blood and lymph. Where they get educated, where they multiply in thymus. But whenever required, they can be sent to tissues. They circulate through blood and lymph. Coming to B cells too have receptors, and they have Y-shaped receptors. Y-shaped receptor. The Y has two, you can see the Y has these two arms and one tail. This tail is embedded inside the B cell, while these two openings, two ends, are open to catch the enemy. This is basically antibody. B cells have receptors which are protein receptors, which are antibodies. Antibodies itself require a single lecture that also I will be delivering. These Y-shaped receptors are present on the surface of B cells, which are also called BCRs. These two circulate in between blood and lymph. B cells need the help of T helper cells. How it happens and when it happens, this requires a different lecture. These all things are interrelated, but to learn or understand it properly, I am dividing into many lectures, short lectures which you can understand and grasp. Now, what happens? These B cells, they differentiate. When they encounter the enemy. They differentiate into plasma cells, and they are stimulated from cytokines produced by T helper cells. So I told you, B cells will take part in protection when they are stimulated by T cells. And how this happens is a very interesting thing. How these T cells goes and tells B cells to get activated, get up and fight. How this happens, this is all very important. And we will enjoy reading it in small lectures. Now, when I said this lecture is confined to granular lymphocytes, so I will just talk about granular lymphocytes. Granular ones, we started with monocytes, then lymphocytes. Lymphocytes are further differentiated on the basis of the enemy they are going to encounter. On that basis, they are differentiated. They are differentiated into B cells, T cells, and they have different roles. Natural killer cells too. Natural killer cells are large granular lymphocytes. Here, they are studied in agranular lymphocytes, but they are activated by lymphocytes. Natural killer cells are activated through lymphocytes and monocytes. It has granules which has enzymes that can kill tumor cells or cells infected with virus. I have already told you that is the role of natural killer cells. But natural killer cells had to be monocytes have to ask them to get activated, stand up. They need monocytes, and they need lymphocytes to be differentiated into natural killer cells. And natural killer cells do have granules, and these granules contain enzymes. These enzymes can kill tumor cells or cells infected with virus. So they control several types of tumors and microbial infections too. This finishes our granular lymphocytes. Thank you for patient hearing. We'll be waiting for your views and reviews. Thank you so much.